Okay, we hit that time limit again, so we're back again for part three now. So again, facilitated diffusion. The term facilitate means to help. So the idea here is that these solutes cannot move across the cell membrane on their own, even though they're still moving along their concentration gradient and do not require energy. They are either too big, they have a charge. Those are the reasons why they can't pass through just in between the phospholipids of the bilayer. So they require proteins. So these are either channel proteins or carrier proteins. So these are those integral proteins or transmembrane proteins. They're the proteins that span the entire cell membrane. They're not just the peripheral ones. So they act like channels or carriers. So again, no energy is required for these. So if we take a look, there's several ways to describe the motion of molecules along these protein channels or carrier proteins. So in these examples here, you can see the phospholipids. These molecules, they're either too big or charged, and they can't just squeeze by here. Maybe they're polar. Again, the interior of here is nonpolar, so they can't get through. They require protein channels or carriers to do it. So these simple diagrams here would be all channel proteins. You're just simply moving along the inner pore of it, which would be a channel. So if you're moving just one molecule, it's called uniport. If it's two molecules and they're both moving in the same direction, it's symport. If they're two molecules moving in opposite directions, it's antiport. So that's some good terminology to know. The channel proteins, these are more for the ions or small polar molecules. And again, it's because the interior is non-polar. So these things can't get by here. They're not attracted to the interior. If something's small, it can squeeze by, like even water, which is a polar molecule, can squeeze by. Oxygen, CO2, they're non-polar. They're small enough to squeeze by, but even non-polar molecules that are bigger, they require a carrier protein or a channel protein. So these channel proteins, you can see, like for example, let's say this is a sodium ion, it's just going through it. It's moving along its concentration gradient. You can see there's more of it outside compared to in. So it's still moving along its concentration gradient. The carrier proteins, a little bit different. The previous example, the protein channel just stayed the same shape the whole time, but these carrier proteins, they tend to change shape. So they have one shape, and when the molecule has a binding spot, once it binds, the configuration of the protein adjusts, and it allows the transport of the molecule to the inside. But again, it's moving along its concentration gradient. So the classic example of channel protein are for ions like sodium ions, and the classic example for facilitated diffusion using carrier proteins would be something like glucose or amino acids. Okay, so now active transport, these are the types of transport that require ATP. So ATP is the energy currency of the cell. We're going to learn a lot more about it in the second unit. It's what's needed to perform reactions or motions within the cell. So they could also be involving proteins. So there's actually two types that we're going to look at that our textbook goes over. And the curriculum asks us to talk about. So that's primary active transport and secondary active transport. There's many different kinds of primary active transport. This sodium-potassium pump is an example of one type, but you can see that a protein is involved. And the big difference between the protein here and the way that it works compared to the facilitated diffusion is that this molecule is required, ATP. The reason it is required is because now you're moving molecules against their concentration gradients. Right? So this is sodium, and the way this works, we'll look at in a little bit more detail in a later part of the course. But the sodium would bind here. There's 3 binding spots, and once it's bound, the configuration of the protein would switch, and now the sodium can be released on the other side, and now there's binding spots that are available for potassium to bind to. It binds to it, and then it flips orientation again, and now the potassium would be released on the inside. So you don't really need to know all the details of how this works, because it does get a little bit complicated, but the idea here is it's going to create an electrochemical gradient because you're moving more positive charge outside the cell compared to positive coming inside because of that 3-to-2 ratio. So it creates an electrochemical